The overall goal of this method is to detect the gene copy number variation in serum or plasma samples.
This method can help answer key questions in advanced prostate cancer field, such as detection of markers useful to predict or monitor response to drugs.
The main advantage of this technique is that it is easy and fast to do. The implications of this technique extend toward therapy of advanced prostate cancer because androgen receptor gene copy number variation is a frequent event.
Generally, individuals new to this method will struggle because the concentration and the quality of cell-free DNA is often lower than expected.
For serum collection, work from 5 milliliter samples of whole blood and serum without anticoagulants, maintained at 4 degrees Celsius. Start by centrifuging the samples for 15 minutes at 1000 G. The supernatant is the serum. Transfer it into 2 milliliter tubes and discard the pellet. Unless the serum is processed immediately, freeze the samples at -80 degrees Celsius. For plasma collection, start with 10 milliliters whole blood samples mixed with EDTA. Then, centrifuge the sample at 1800 G for 15 minutes, at room temperature with no break setting on the centrifuge. To reduce the chance of contamination, only collect the upper part of the